Diabetes mellitus is a devastating chronic disease characterized by elevated glucose level in the blood that affects up to 10% of the Western population, and it's more than 20% of the population in some developing countries like Pacific Islands and Middle East. Individuals with diabetes have devastating complications, including gastrointestinal disorders, which can be present in up to 80% of individuals with diabetes. Gastrointestinal symptoms may include difficult digestion, malabsorption, pain, diarrhea, leading to frequent hospitalizations. Currently, there are no therapies available to prevent the onset of gastrointestinal disorders, and investigators believe that these symptoms may be related to some sort of intestinal stem cell abnormalities. Studying individuals affected by type 1 diabetes, which is a juvenile form of diabetes, we discovered that the levels of a protein called IGF-BP3, or insulin growth factor binding protein 3, are abnormal in type 1 individuals with gastrointestinal symptoms. Furthermore, we discovered that IGF-BP3 is capable of destroying the colonic stem cell in vitro and in mice once injected in vivo. Indeed, IGF-BP3 inhibits the growth of stem cell-derived organoid in vitro by binding its receptor TMEM219, which we discover is expressed on colonic stem cell. We trust that we have discovered a novel function for the hepatic-produced protein IGF-BP3, or the hormone controlling the colonic stem cells. This animation depicted the increased production and release of IGF-BP3 by hepatocytes during hyperglycemia, and how circulating IGF-BP3 may bind either to IGF-I, thus preventing its basal positive pro-survival tone on colonic stem cells, or to TMEM219, thus activating caspase-associated pathways and leading to colonic stem cell death. Interestingly, we have cloned and produced the extracellular domain of the TMEM219 receptor, thus creating an extra body, and used this recombinant soluble human ecto-TMEM219 to quench the elevated IGF-BP3 in the blood. The injection of the novel ecto-TMEM219 in vivo in diabetic mice prevented the diabetes-associated high glucose-mediated destruction of colonic stem cell in vivo, thus preventing the onset of intestinal disease and re-establishing the homeostasis of colonic stem cells. The discovery of what we called enterostaminin, or IGF-BP3, as a new hormone that can control the growth and development of colonic stem cells is an extraordinary achievement that could open unexpected scenarios in the field of regenerative medicine and diabetes. In addition, this work will change and reshape the way of designing stem cell therapy and, in general, the current thinking on how stem cells should be used for various diseases. The inhibition or stimulation of hormones that control stem cells will allow to fully exploit the regenerative capabilities of stem cells. I think that many other hormones with similar functions will be discovered.